Vitamin D's relationship with immune function has been studied intensively over the past 2 decades, and the research has consistently pointed toward a more mechanistically specific role than simply supporting immune health. The vague language that appears on supplement labels without explaining what is actually happening at the cellular level. What vitamin D does in immune cells through the vitamin D receptor is directly activate the genes that enable macrophages and other immune cells to kill pathogens that have taken refuge inside them. That intracellular killing mechanism is autophagy, and understanding this connection changes how we think about vitamin D deficiency and susceptibility to infection. When calcitriol, the biologically active form of vitamin D, binds to VDR in macrophage nuclei, it upregulates a specific set of autophagy genes including ATG5, ATG16L1, and BECLIN1. These are not genes with secondary involvement in immune function. They are the structural components of the autophagy machinery that engulfs and degrades intracellular pathogens in a process called xenophagy. The most extensively studied application of this mechanism is tuberculosis resistance. Mycobacterium tuberculosis has evolved strategies to survive inside macrophages by preventing lysosomal fusion. Vitamin D-activated autophagy overcomes this resistance by forcing lysosomal degradation of the pathogen through an alternative route. This mechanistic pathway, characterized in 2006 research, was one of the more significant findings in innate immunity that year and provides a molecular explanation for the historical observation that sunlight and sanatorium treatment improved tuberculosis outcomes. The neurological dimension of VDR-driven autophagy adds another layer of clinical relevance. Vitamin D receptors are expressed throughout the brain, and VDR-dependent autophagy induction in neurons contributes to the clearance of amyloid beta and tau protein aggregates, the pathological hallmarks of Alzheimer's disease. Population studies consistently find associations between lower vitamin D status and higher dementia incidence, and while establishing causality from observational data is always appropriately cautious, the mechanistic pathway through autophagy-dependent protein aggregate clearance provides biological plausibility for the association. Optimizing vitamin D for autophagy-supportive immune function involves targeting serum 25-hydroxyvitamin D levels of 40 to 60 nanograms per milliliter, a range that requires 2,000 to 5,000 IU of supplemental D3 daily for many adults in northern latitudes, particularly during winter months. Sunlight exposure of 10 to 30 minutes at midday during peak UV seasons can maintain adequate status for people with lighter skin tones in temperate climates, but this window narrows considerably above 35 degrees latitude in winter and for individuals with darker pigmentation who require longer sun exposure to produce equivalent vitamin D. Dietary sources: wild salmon at approximately 988 IU per serving, sardines at 193 IU, contribute meaningfully but rarely suffice as sole sources without supplementation. Magnesium deserves co-acknowledgement because it is required for the enzymatic conversion of vitamin D to its active form, and magnesium deficiency, prevalent in populations eating processed food diets, can impair vitamin D activation regardless of how much is supplemented. Vitamin K2 functions as a complementary partner directing the calcium mobilization that activated vitamin D initiates, and the combination of D3 with K2 supplementation is the current standard of informed supplementation practice for people targeting cardiovascular and immune autophagy benefits.